In today's lecture, we're going to talk about recombination and linkage mapping. Recombination occurs during meiosis, and it's the result of something that happens when you have two genes that are linked, but you get switching of information between homologous chromosomes, between the maternal and the paternal chromosomes. So let's take an example here. So here's an individual that is big A, big A, big B, big B. Here's an individual who's little a, little b, little b. You can see this is the only possible gamete this individual can produce. This is the only possible gamete this individual can produce. These are the F1 individuals you'll get. All right, so you're with me so far. But then I want to show you this. What happens is when this individual is producing gametes, if A and B are linked, they're on the same chromosome, and you can see if big A is with big B and little a is with little b, these are the only two possible gametes they can produce. These are the non-recombinant. In other words, big A is stuck with big B, little a is stuck with little b. They're the only ones you can get. On the other hand, if crossing over happens, and only if it happens, then you can get these combinations of gametes. You can produce novel ones. It's a very interesting thing called recombination, and you'll see later in this lecture we can use that to create a genetic map to see how far apart different genes are located on a given chromosome. So some terms you have to know in this lecture whenever we're talking about recombination, just to understand the differences. So one term is input gametes. These are the gametes that are used to make an individual. So if you think of yourself, these would be your mother's egg and your father's sperm. Output gametes are the gametes made by the individual. So again, if you think of yourself, these are your eggs or your sperm. Recombinant gametes are output gametes that are different from input gametes. Sort of makes sense, just the process that we talked about. And then recombinant individuals are individuals that are made from recombinant gametes. Now you might be thinking this, am I a recombinant individual or am I non-recombinant? Well, keep in mind, it depends on what genes we're talking about. All of us are going to be recombinant for some genes, right, of the thousands of genes we have. Others of us will be, the same person will be non-recombinant for other genes. So you're recombinant and recombinant, but it depends what genes we're talking about. So keep that in mind. Okay, so what happens when two genes are on the same chromosome? So let's consider this scenario here. Let's say that we have an organism that has purple flowers and long pollen. Big P encodes for purple, little p encodes for red, big P is dominant to little p, and we're assuming true breeding strains to start with. Right? So the usual spiel that we use in genetics. This is a long pollen. Let's say big L encodes for long pollen, little l encodes for short pollen, and again, big L is dominant to little l, and we assume true breeding. So we assume all that. These are the genotypes that would happen. Big P, big L, little p, little L, little l. This is the situation, but look at the genes are linked. P and L are on the same chromosome. So if that happens and these two flowers mate and they have offspring, you can see that this is going to be the only type of offspring that can be produced. So it's going to be big P, big L, little l, but the big P and big L are on the same chromosomes. So if we take that individual again, or great genetics, please do it twice, right? And you can see that here we're mating this individual with this individual, the same ones. And if we produce these four individuals on the bottom, these would be the genotypes that are associated with them. The question then becomes, which of these four are recombinant, which are non-recombinant? Two ways of telling. If you can grab one chromosome from this individual, and make one of these four offspring, then they're non-recombinant. So if you look carefully, go ahead and look, which of the four are non-recombinant? Pause the video if you have to. Okay, so let's look at the answers. So you'll see that this individual is non-recombinant and the last individual is non-recombinant because it's possible to grab one chromosome from each and make those individuals. In other words, let's take the little one. That's the easiest one. To make that, we would grab this chromosome and we would grab this chromosome, right? Boom, we made that person. This one here, we would grab this chromosome, and then it's saying it doesn't matter what the other alleles are, so we can grab either of these. Which of these four are non-recombinant? Or excuse me, we just did that. Which of these four are recombinant? Okay, obviously it's the remaining two, right? But why? This is this individual's genotype. This genotype belongs to this individual. And you can see that for each of these, we cannot grab one chromosome from here and one chromosome from here and make them. Let me show you what I mean. Let's look at this one. Big little p. So to get little p, we have to grab this one and we have to grab this one. So we got the little p. But notice that if we grab a little p and a little p, what comes with them? Little l, little l. There's a big L here, there's no little l, so we couldn't have made that individual. So it's considered recombinant. It had to have been made from crossing over happening. Another way to tell is that the recombinant individuals are often underrepresented. See how their numbers are lower? The non-recombinant have higher numbers of offspring. So there's two ways to tell. Okay, so given the below cross, which of the following offspring are considered recombinant? Right, which of those are recombinant? Again, it would be number 3, right? Little p, you can't have a big L in there. Okay, so how does any recombination occur when genes are on the same chromosome? Well, it happens through crossing over in prophase 1, right? And specifically, the main time we have that is pachytene. So this slide is really just sort of linking different concepts from different lectures, relating this to the lecture on meiosis that you had watched previously. Okay, let's apply this now. So we talked about recombination, we talked about how it works. Let's apply it to something concrete. Why would we use this? So we'd use this for a couple of reasons. One is we can use it to test for linkage. In other words, we can test to see if two genes are actually on the same chromosome or not. And we could do this, we could do this by mating an unknown individual with a known homozygous recessive individual. And if we do that, we can tell if two genes are linked or not. So what happens here is if we know this individual is big M, big D, little d, right? Big M, big D, little d, we know that, that's their genotype, but we don't know if they're this, if the genes are linked, or if they're this, the genes are not linked. So to figure that out, what we're going to do is we're going to mate them with a known homozygous recessive individual for each gene. So little m, little d, little d, little m, little d, little d. The reason we do that, is because you could see that the homozygous recessive individual contributes nothing in terms of diversity. Now they're contributing something, right, egg or sperm, but they're contributing nothing otherwise. In other words, they're always going to give little m, little d in each situation, so you know that's all they can contribute. And if you know that, it makes the problem a little bit easier. You know any diversity isn't coming from them, right, but it's coming from this other individual. If two genes are linked and they're completely linked, you can see that only two gametes can be produced, big M, big D, and little m, little d. So if you mate this with this and this with this, these are the two individuals you get. So in this cross, if you get two phenotypes, you know the genes are completely linked. On the other hand, if you do this test cross and you get 4 phenotypes, you know the genes were not linked, or they were incompletely linked, right? So there's some crossing over happening at least. Because you could see that we get 4 gametes here, right? We foil, we get 4 gametes. And you can see that since there's 4 gametes here, 1 here, 4 by 1 Punnett square, we're going to get 4 phenotypes. So one thing I want to mention, just to keep in mind, is that there's 3 categories, right? There's complete independent assortment, where the genes are not on the same chromosome, and the results would look like this. There is linkage when there's incomplete linkage. In other words, for incomplete linkage, what we mean is the genes are on the same chromosome, but some crossing over is happening. So you will see some recombinant offspring. So you will see 4 categories, but these would be underrepresented. The final category is complete linkage, and that's this scenario. You're only going to see 2 categories, and that's it. Okay, your book will say something like this. I just want to point it out, that a single crossover produces half non-recombinant gametes, half recombinant. All they mean is this. So this is no crossing over. It's your control group at the top, not much to talk about. The bottom we have crossing over, we have 2 homologous chromosomes. It's saying if this sister chromatid crosses with this one, right, so we get crossing over these 2 right here, we'll get these recombinant gametes. So all it's saying is we have 1 crossover event, that's what this is, and so half our gametes are recombinant, and the outer 2, right, the other half are non-recombinant. That's all this slide is saying. Okay, we can use a test cross to test for linkage, right, and we can use it to calculate the distance between 2 map units. Sorry, the distance between 2 genes in terms of map units. And so the formula to do this is this. So if you want to calculate the recombination frequency, it's the first step. What you want to do is you want to take the number of recombinant progeny, which on this slide is what, 8 plus 7, that's 15, divided by the total number of progeny. So you have to add all these up, right? So what is this? This is 55. This is what, 108 plus 15. 15 is what, 123, okay? So 15 divided by 123 times 100, and that's going to give you something called the recombination frequency. So which of those would it be here? If you add that up and you sort of guesstimate, it'd probably be number 4, right? 12.2% recombination frequency, or percent recombination. The nice thing here is that when you get this percent can be directly translated to something called a map unit. So if you have 12.2% recombination between genes M and D, what that means is the map units between the two genes are 12.2 map units. Helps you basically measure the distance between genes in a relative sense, and we'll talk about that in a second. Okay, some other terms that you want to know. Some other terms that you want to see is that the way alleles are arranged on the gene, we have to note that somehow, right? So if you have an individual who's big A, big B, little b, this is also big A, big B, little b, right? But they're two very different scenarios. So if the genes are linked, you got to say, is it cis dihybrid or coupling? In other words, are the dominant alleles on the same chromosome? Or is it trans dihybrid or repulsion? Are the dominant alleles kitty corner from each other, right, on different chromosomes? The reason you have to say that is because it has extreme implications on which gametes are recombinant, which are non-recombinant. So in other words, the non-recombinant gametes here would be recombinant gametes for this individual, right? So it's sort of the opposite sides of the same coin. So what I would say on this slide, practice a little. Draw all the possible gametes and indicate which ones are recombinant. See if you can do that exercise. Some other terms that you want to know. You want to know what interchromosomal recombination means, and this is basically a synonym with the phrase independent assortment. You also want to know what intrachromosomal recombination is. Again, this is just a synonym or a fancy way of saying crossing over. I point these other terms out just because people will use them sometimes in conversation, but they mean the exact same thing as the term that's right after them in parentheses. Okay, so let's talk about the final thing here is how to make a linkage map. How do we make a genetic map versus a physical map? I like this quote. I think it's sort of nice. So a famous scientist in the early 1900s said, I went home and spent most of the night to the neglect of my undergraduate homework, right, in producing the first chromosome map. So I usually neglect my undergraduate homework and watch TV. But he did something slightly more productive, which is nice. And what he did was he produced a something called a linkage map. It's very interesting. And so he did that by analyzing the frequency of recombination between genes, and he predicted the relative location of the genes on a chromosome. So I want to really make sure you understand that key word here is relative. I would circle that word. In other words, if you have a genetic map, what it's saying is this. Let's pretend I'm walking across the screen. So my pointer's walking across the screen, right? Walking. How far did I walk? If it's a genetic map, and I walked from here to here, I walked halfway across the screen. See, that's a relative term. I don't know how far halfway is, right? But I know it's halfway. That's a genetic map. If I walk again, and it's a physical map, I literally have to measure and say, maybe I walk 15 centimeters across the screen. So you sort of get the idea. So genetic map and physical map both measure distances, but genetic map is just relative. And that's what we're producing here when we talk about linkage mapping. So how does this work? So basically we use that formula we thought of before, right, that we mentioned a few slides ago, and we figure out the map unit distance between two genes. If we have a bunch of genes in the problem, it's sort of plug and chug, and you just have to say relative to each other, what are the distances, and then you can actually get the distances between genes for crosses you hadn't even done, if you fill in enough of the gaps. So I'm going to sort of walk you through this a little bit. So I have a bunch of pairs here, right? A and D, B and D, and C and D. I have recombination frequencies that we calculated. So those are the same thing as map units. When I'm making a map, I start with the larger value first. So I say there's 23 map units between C and D. I say between B and D, there's 13. And I say between A and D, there's 8. If I do all this, map units are additive, at least over shorter distances. They're additive. So in other words, if this is 13, this is 8, I can assume between A and B is 5. So see how they're additive. I can assume that. If this is 23 and this is 13, I can assume that B and C is 10 because they're additive. So you can see how I'm getting distances between genes that aren't even listed up here, right? If I plug and chug and put them together. Now these genetic maps are nice in the sense that if you're wondering if you answered a question correctly, if the map you have matches all these statements and they're all true still, you know you cannot be wrong. So that's sort of nice. The hard thing is there's no really rhyme or reason in terms of putting them in order. I start with the bigger numbers, work my way smaller, but it's really trial or error. So when you construct these maps, you might find yourself taking a pencil and erasing them and then doing them again. The other thing I want to note too is that some maps might not be the only way to satisfy a problem. So in a sense, this isn't a good problem I gave you, because why does gene A have to be 8 map units to the right of D? If I just swiveled it around, took A, pulled it over here, couldn't it be 8 map units to the left of D? Everything would still be true, right? So you can see that this isn't a good map because there's not enough contingencies on here. But you get the idea. Sort of another map that you could just, again, try to put together, right? So the thing we talked about on the last slide, it's nice to sort of put it together, see if you can analyze the different distances. I'd encourage you to make up some maps on your own, see if you can put them together and figure out the distances. Okay, the final thing I want to walk through is sort of a real problem, right? This would be a good practice problem for you too. So I'm going to give you the answer on the next slide, but I encourage you to practice this problem over and over, not having the answer in front of you. See if you can do it by hand without a calculator. And so this is just a problem, again, looking at linkage between two genes, right? So PR for purple eyes, VG for vestigial wings, right? And you could see here that, it's common fruit fly nomenclature, right? So if you don't have purple eyes, it's PR plus. If you don't have vestigial wings, it's VG plus. Walk this all the way through, taking these observed values here, figuring out who's recombinant and who's not. I want you to go ahead and see if you can calculate this map unit distance of 10.7. Some final statements that we want to note here about this lecture. So I said that map units are additive, and they are, but the exception is if you get very, very far distances, so distances that are more than 50 map units apart. Basically, you're more likely to have multiple crossing overs happen where the genes cross over, cross back again, and you're not seeing it in real time, right? You're just seeing the result at the end. You're seeing where the alleles are located at the end. You're not seeing how many times they crossed over. So if you have very, very large distances, what happens is the map units can be underestimated, and so that additive rule doesn't quite apply anymore. Okay, just a summary of things we talked about. Encourage you to look this lecture over again. Any questions you have, please let me know.